Hi, Jonas. Thank you very much for taking the time to do an interview update for Open Medicine Foundation and the audience here today.
Hi, Chris.
Hello. Obviously it's been a very crazy year this past year, but I thought maybe we should start with a very recent paper that you had published in regards to a hypothesis relating to ICU and recovery and how that looks to be a similar mechanism, or you hypothesize to be a similar mechanism to ME/CFS.
Yeah, that's a paper we published in Frontiers in Medicine recently, and we are there trying to allude to that the symptoms and the situation we see in intensive care unit patients that have been critically ill and been through quite severe medical treatment also, in some cases end up with symptoms that are quite similar to what we see in ME/CFS patients. And now we are trying to learn from these mechanisms that we have been studying quite well in intensive care unit patients. So to make the story a bit possible to grasp, I think we should maybe just focus on a few things that we are looking at. There's plenty of information in that paper. So as you all know, probably we have a lot of different endocrine glands in the body, endocrine organs that signals to the rest of the organs in the body and also controls a lot of our biological functions in the body. And what we know is that some of these adrenal glands and pituitary glands and hypothalamus, etc., parts of the central nervous system will be affected during critical illness, and they will stop sending out these signals that we need to actually regulate our normal functions in the body, or they will not have the rhythm that these endocrine organs need to have to actually control our normal functions. So our idea is now to try to see if we see the same pattern actually in our ME/CFS patients. So initially what we will focus on is hormones like the steroid hormones, sex hormones, thyroid hormones, and some of the neurotransmitters that are also released by these adrenal glands and pituitary gland and hypothalamus. So I think we can actually learn something from this and use the similarities, but also the differences we see between critical ill, intensive care unit critical ill patients and the ME/CFS patients.
Yeah, that's fascinating, and this is the type of research that we really want to kind of see transition to ME/CFS, taking it from other diseases or other types of illnesses, other mechanisms, and seeing how that could relate to this disease. And obviously, it's very relevant at this time as we speak of the COVID pandemic and all sorts of different types of illnesses that are coming from that, the long COVID. And how that relates to ME/CFS. Obviously, Open Medicine Foundation has recently funded research into looking into COVID-2-ME, and I know a large majority of that work is happening there in Sweden, or at least a large majority of the collection of samples. I thought maybe it'd be great if you could kind of bring the audience up to speed on that.
Yes. So what we are doing now, as a cause of the pandemic, actually, we have a unique opportunity to actually collect and follow patients where we have a known onset of an infectious disease, a viral onset, and we are now able to monitor over time what happens in those patients. And as we already now see, now it's 1 year since the pandemic hit Sweden. I think we have now a chance to actually see how patients are going to react and what kind of symptoms that will be long-lasting after the infectious disease in some of the patients. Not in all, of course, but in a quite large percentage of the patients. So what we try to learn now is when we know we have an ongoing a clear onset of a disease, like an infectious disease, what happens in the body? How does the body cope with the situation? And when it comes to COVID, we know that some patients will have neuroinflammation. So actually, even viruses will transport into the central nervous system and also get quite severe neuro related onsets. And some of these patients end up in our neurointensive care unit for treatment. While others have so much milder symptoms, they don't get, they feel they are sick, but they have like a mild flu maybe. And still, when they have recovered from that initial infection, they still have ongoing sequelae, as we say, symptoms afterwards. And those are, to a large extent, very similar to what we see in our ME/CFS patients. So this is what we're trying to use now as a, sort of unfortunate situation, but also a possibility for us to actually see and follow on site what is going on in both periphery and the central nervous system. So in parallel with that, if I may, in parallel with that, we have also an opportunity to monitor another patient group that we have had an opportunity to look at, and that's patients with herpes simplex encephalopathies. So herpes virus initiates in some patients a very severe degradation and inflammation in the central nervous system. And we now are monitoring a cohort, a fairly large cohort that's been collected over time, where we have multiple sampling points over the disease and when the patients recover. So we have now cerebrospinal fluid, the liquid that is surrounding the central nervous system, and we also have plasma blood samples collected over time in these patients. So what we're doing there, we are doing the same thing as we're doing on our post-COVID infected patients and our ME/CFS patients. We do proteomics, deep screening of all the protein biomarkers we can find in the body fluids. And in parallel, we do metabolomics, which is the small molecular patterns that are arising from the metabolism in the patients. And from there, we hope to learn what are the signaling mechanisms, what are the biochemistry behind the situation when patients are recovering from the disease, and also maybe learn about potential treatments.
I mean, I think one of the things that I like the most about this whole setup of research that you're doing there, Jonas, is the fact that you're looking at, ME/CFS, you're looking at the transition of COVID to ME, or what other people might be calling long COVID, and you're looking at this transition from HSE to these ME-like illnesses. And I think the benefit of that really is that you're going to be able to identify that whatever this ME/CFS signature kind of transitions to from these other diseases, there may be a commonality there, which may provide a good line of evidence to suggest that regardless of the trigger, regardless of COVID, regardless of HSE, these people end up looking like the same signatures as you see in ME/CFS. And that's something that I'm kind of looking forward to seeing how that kind of plays out.
Mm. No, I agree. And maybe I can give you a giveaway already that we are actually now, hopefully soon, publishing a follow-up paper also in Frontier Medicine, where we will actually allude to some of the treatments that has been used over years in intensive care unit patients, for instance. And we are trying to see what kind of mechanism that those therapies are using, and see if those will be relevant also for ME/CFS. And I think that's where we want to go in the end. We want to figure out what is going on, what is the biochemistry, what is the pathophysiology in the patients, but also we have to come to a treatment. And I think that is our goal, really.
Maybe I thought it'd be nice to kind of spend like a minute maybe updating where along. I mean, obviously you've described the projects that are ongoing, but whereabouts are you at this stage with the COVID to ME projects and with the HSE, following the HSE projects as well?
So with the COVID studies, we are continuously collecting. So we are trying to collect as much material as possible now and get this temporary resolved sampling going, so we can follow the same individual over time, which I think is crucial. But we are doing currently proteomics in the patients where we have cerebrospinal fluid, that's liquid around the brain. We do deep proteomics. So we try to follow all the protein biomarkers that we can monitor with for looking for neuroinflammation, looking for cell damage, cell repair systems that are very relevant for this patient group. And in parallel, we do this metabolomics, the low molecular weight screening, both in cerebrospinal fluid, but also in blood samples that we are collected. And with the HSE patients, we are already done with all the proteomics. So we're already done, and we're actually doing the final data evaluation now, which is very intriguing and also very complex, as you may understand.
Yeah.
So, thousands of proteins that are going to be measured and correlated to clinical situation. And we're also right now doing the metabolomics. So we will be able to compare also what the protein picture is and how the metabolism is related, both to the clinical situation, but also to each other, how proteins are regulating metabolism and metabolism potentially regulates proteins also. So it will be a very unique possibility here.
Yeah, I mean, it would be interesting as well to seeing the relationship between cerebrospinal fluid and the blood plasma metabolites and proteins as well, as you're looking at that. It sounds like obviously the HSC work may be coming out, sometime in the very near future, and the COVID research is also underway with the assays occurring as we speak. So that's very exciting news. And it sounds like you've been pushing pretty well ahead there in Sweden over the past year. So, I mean, that's fantastic to hear.
And also, we have, of course, another track that we are following in parallel. Everything has to go in parallel here to keep up the speed, basically. As some of you know, we published a paper last year also on the autoimmunity and the auto antibodies that we have monitored together with Carmen Scheibenbochen in Berlin in our Swedish cohorts in ME/CFS. So actually what we are doing in parallel now also is to see if we see the same kind of pattern in early stage of patients with COVID or with HSE, actually, to see if it is one part of the explanation could be that you cause onset of inflammation due to the initial viral infection. And as a side effect, a negative side effect, you get some sort of autoimmune reaction. And in some patients that may end up with an autoimmune antibody that may cause problems. So I think we have that we haven't dropped that idea yet. So I'm looking into that in great detail right now.
Certainly. Do you see any point in the future where that type of work can kind of come together with the work that you're doing with HSC and COVID or anything like that?
Yeah, definitely. So we try to learn from the different materials we are collecting. And I think we can, since we know it's the viral onset in HSC and COVID, and to a large extent, we know that many of our ME/CFS patients present with an initial infection when they debut in their disease. So I think we can learn a lot from these parallel tracks.
Certainly. I think it'd be interesting to know, at what point along the line that autoantibodies start to show up, autoimmunity starts to show up within these patients, whether it's quite early on or whether it's something that happens far along into the disease of ME/CFS. So I think there's plenty of possibilities happening over there in Sweden. Yeah, I mean, it's been fantastic to hear about all this type of research that's going on. And I think obviously you must be pretty excited with all these things that are coming out this year. Is there anything else that you're particularly excited or interested in explaining to the people at home?
I think we have there's a lot of things going on. So one thing that our PhD student is working intensively with is the method development. And of course, what we need to do in this field where we are sort of pushing the limits for technologies and measurements, we always need to improve the methodology. And that is ongoing work, and it will be continuous work, I would say, during the whole PhD program for our students. But I think it's very important to understand that there is limitations in all technologies, and we have to improve them to make them more accurate, more precise, giving the right information because the methods, the measurement methods are sort of limiting what we can see, and there's also biasing what we can see, so to say. I think that's a very important task for us.
Yeah, I think it's probably like less exciting, but it's definitely very important, I think, in some of the research concepts, and I appreciate that a lot. I know that you've been working on certain panels in pathways that are probably of great interest to ME/CFS within that, and trying to develop methods for that. So, you know, it was interesting to listen to that. And I know you were developing methods, I guess, to measure these hormones. And so that's something, as an example, that you've recently developed that method, and now you're able to look into that hormone work a lot more thoroughly. And that pertains, I guess, to the initial comments you made at the start of this talk in regards to your hypothesis and how the hormones relating to this inflammation system. So this is how everything kind of continues and gets started, yeah, you can see kind of science in motion. I think, yeah, in terms of like bringing it back to that hypothesis, because I thought there was some interesting aspects as it related to how the hormones are related to the neuroinflammation, and there was a nice little cycle that you had, a simplification cycle, I think, that involved, you know, the hormones relating to oxidative stress and inflammation markers. And a few of those oxidative stress things were nitrosative stress as well, which is probably something that a number of people have probably read in the literature in relationship to ME/CFS before. So I thought that was interesting that you were touching on a few of these common things that we've seen in this disease as well.
No, definitely. And I think there's, of course, a lot more to do there to understand its very complex regulatory mechanisms. But yeah, one of my tracks is, of course, the neuroinflammatory part. And I think if we can understand why this low-grade neuroinflammation seems to occur and what is the reason for that, in combination with lack of energy and oxidative stress and nitrosative signaling, etc., etc. I think we are on to something there. I think difficulty to measure sometimes, but I think we have to do our best. I would like to add also, what is important also, and that's we are also spending quite a lot of time on right now, is when it comes to methodologies and measurements that we are combining and also comparing different measurement tools. So I think that is also very important, to not only to validate methods to each other, but also to complement each other. So we get certain possibilities with certain methodologies, and other methodologies will maybe very sharp on certain things, but lack the sensitivity for some of the targets that we have. So I think a combination is very important, and collaboration in that sense also extremely important.
I would definitely agree with that. And I mean, obviously some of the things that I think are really interesting to me is, I mean, obviously in this hypothesis you described it as a circle, and I think that's an important aspect to understand, that when we're looking at all these different pathways, that it really is kind of like a cycle, and it's not always trying to look for cause and effect. The cycle is, I guess, the thing that we're trying to look at in its entirety. And then looking at these kind of repeated measure studies, or these longitudinal studies in regards to, or these cohort studies where you're looking at these diseases and how they may transition to different types of recovery. I think that's an area where we could potentially start looking into this cause and effect aspect as it pertains to how a cycle can kind of transition and change over time. So yeah, I mean, the culmination of everything that you're doing is really, really interesting. I think there's going to be it's an excellent way of looking into ME/CFS and understanding it better. And I have a lot of hope for the work that you're coming out with at the moment and into the future. So thank you very much for taking the time, I think, to speak to everybody about this. It was very much appreciated, and we'll catch up soon.
Thank you. All the best to all of you.